Assalamu alaikum and good day. We are going to look at irreversible cell injury and tissue degeneration in this lecture.
In this lesson, we will describe irreversible cell injury, list the different types of necrosis and their morphological changes, describe cell death due to apoptosis and its difference from necrosis, and list differences between dystrophic and metastatic calcification.
From the previous lecture, we know that when there is irreversible cell injury, this could lead to cell death. And there are two types of cell death that we encounter: necrosis, which is defined as death of group of cells or tissues in a living person. There's also apoptosis, which is a programmed cell death. That is a specific type of cell death involving single cell or a small group of cells in the tissue where the other cells are functioning normally.
There are other mechanisms of cell death apart from necrosis and apoptosis. Necroptosis is a hybrid between necrosis and apoptosis, whereby it resembles necrosis morphologically, but it is genetically controlled form of cell death, just like apoptosis. Pyroptosis is a form of apoptosis that is accompanied by fever-inducing cytokine interleukin-1. And ferroptosis is a distinct form of cell death that is triggered when excessive intracellular levels of iron or reactive oxygen species overwhelm the glutathione-dependent antioxidant defense that cause unchecked membrane lipid peroxidation.
Irreversible cell injury occurs if the cause of the cell injury persists and the cell reaches the threshold where it is no longer reversible. So the features that we see are membrane damage, influx of extracellular calcium and the release of intracellular calcium, lysosomal swelling, mitochondrial vacuolization, and pycnosis, karyolysis or karyorexis of the nucleus. There are two major mechanisms involved in irreversible cell injury. The first one is mitochondrial dysfunction, and the second one is membrane function disorders. Membrane function disorders is the most central factor in the pathogenesis of irreversible cell injury. Now we look at the mechanism of irreversible cell injury, necrosis due to ischemia. Two major mechanisms for cell injury are mitochondrial damage and membrane injury. We concentrate on the membrane injury. The membrane injury, when there is membrane injury, there's going to be leakage of enzymes and also increase in calcium influx, and that would lead to many other damaging factors in the cell that leads to cell death. Now we look at the different types of necrosis and its morphological changes. Necrosis is death of group of cells or tissues in a living person. Necrosis refers to a sequence of morphologic changes that follow cell death. There are 6 types of necrosis that we will see. Coagulative necrosis, which is associated with ischemia. Liquefactive necrosis, which is associated with presence of hydrolase enzyme. Fat necrosis, which can be enzymatic or non-enzymatic. Caseous necrosis, which is associated with tuberculosis. Gangrenous necrosis, which is associated with ischemia on top of bacterial liquefaction, and fibrinoid necrosis, which is seen in immune reactions. The morphologic appearance of necrosis is a result of 2 processes: denaturation of proteins and enzymatic digestion of the cell, and it can be either one or both of these processes together. Microscopically, dead cells are more eosinophilic. The cytoplasm becomes vacuolated and the cell may become calcified. The nucleus becomes either pycnotic, karyorectic, or undergo karyolysis. This is what we mean by karyolysis. The nucleus, which is supposed to be very dark, becomes faded in karyolysis. When the nucleus undergoes pycnosis, it shrinks into half or more than half of its size. And in karyorexis, we see that the nucleus becomes fragmented. So these are the features that we see in a necrotic cell. Microscopically, we will see karyolysis, where the nucleus is faded, and karyorexis, when the nucleus are fragmented. Compare this picture of necrotic tissue with the normal cells. The normal cells have very dark and elongated nucleus, like these ones, and they are faded in karyolysis and fragmented in karyorexis. This picture shows the nuclear feature of pycnosis. You can compare the normal size of the nucleus with this shrunken nuclei that has undergone pycnosis. Okay, so this area are the necrotic area, and these are the remnants of the normal tissue. First, we look at coagulative necrosis. The cause of coagulative necrosis is ischemic hypoxic injury. The picture that we'll see is infarction, which is defined as tissue death caused by a local lack of oxygen due to an obstruction of the tissue's blood supply. Coagulation of the tissue is caused by protein and enzyme denaturation due to increased acidosis. Remember, we discussed about in hypoxia, there is increased lactic acid production due to production of ATP via the anaerobic glycolysis pathway instead of the normal oxidative phosphorylation, which occurs when their oxygen is abundant. The general architecture of the tissue is preserved because of the denaturation of enzymes. Denatured enzymes are inactivated enzyme, so it cannot break down protein and tissue. This feature is seen in most organs, especially the heart, the muscle, the spleen, and the gut. This picture shows the coagulative effect on tissue as displayed in an egg. The dead tissue or egg retains its shape and cell outline even though after it's being poached in hot water because of inactivation of the enzyme. This picture showed an area of necrotic tissue in the heart, as pointed by the arrow in the first picture. Under the microscope, this necrotic area is represented by the darker pink cells. Compare the necrotic area with the normal tissue around it. These are the normal area. Note that the cells still retain its cellular outline even though it's necrotic. So this is one cellular outline, another cellular outline. Okay. Note also that the nucleus in the dead cells are either smaller than the normal nucleus or the nucleus is absent. So the nucleus has undergone karyolysis or pycnosis. Next, we look at liquefactive necrosis, which basically means the dead tissue becomes liquid instead of solid state. This type of necrosis is seen in two types of conditions, in the brain and in bacterial infection. Liquefactive necrosis in brain cells. The brain cells are rich in hydrolytic enzymes and lipids and has very little connective tissue. When the brain cells becomes necrotic, for example in stroke, where there is obstruction of the blood supply to the brain, the cells are digested by their own hydrolase. The brain tissue becomes soft, liquefies, and is walled off from the healthy tissue, and this forms a cyst. Liquefactive necrosis can also be caused by bacterial infection, for example, Staphylococcus, Streptococcus, or Escherichia coli. In bacterial infection, neutrophils release enzymes to destroy bacteria. These enzymes also destroys the tissue and cause it to become liquefied. This picture shows a brain tissue from a patient who had a stroke caused by obstruction of the blood supply to the brain. Compare the area of the brain at the arrow with the normal brain around it. One area shows cystic space and the tissue becomes liquefied instead of in solid form. This can also be seen under the microscope on your right. Compare the necrotic tissue pointed by the arrow with the normal tissue around it. The tissue loses its normal architecture and loses the normal cellular outline. Compare that with coagulative necrosis. Next, we look at fat necrosis. This type of necrosis is seen in two types of condition: in breast, which is usually due to trauma, and in pancreas, which is due to the enzyme action to the pancreas. We look at the enzymatic fat necrosis in pancreas first. The pancreas is rich in enzymes, and one of the enzymes that it produces is lipase. The pancreas itself is composed of a lot of fatty tissue. When there is injury to the pancreas, the enzymes of the pancreas leaks out and the fat tissue is digested by lipase. The lipase breaks down the triglycerides, releasing fatty acids which can combine with minerals such as calcium, magnesium, and sodium ions, and they can make soaps. The necrotic pancreas tissue will look chalky white. Another condition where fat necrosis occurs is in the breast. The breast is also rich in fat tissue. After trauma to the breast, hemorrhage can occur. The hemorrhage causes swelling of the breast tissue, which can cause increase in pressure in the breast tissue, pushing the blood vessel to collapse, leading to ischemia and pressure necrosis. The net effect is fat necrosis. Look at this picture. The patient had acute pancreatitis, which causes the lipase enzyme from the pancreas to leak into the surrounding tissue. The fatty acid released by the fat digestion binds with calcium and other minerals to form soap, which can be seen as the white areas over the pancreas. Fat necrosis is seen under the microscope in this picture. The cellular outline is retained. You can see the cellular outline of one cell here and another cell here. But the fat cells have lost their nuclei, and the cytoplasm has become this pink amorphous mass of necrotic tissue. Now we look at caseous necrosis. Caseous means cheese. This refers to the cheese-like appearance of the yellowish necrotic area on gross examination, just like the one seen in this picture. It is characteristic of tuberculous infection, which is caused by Mycobacterium tuberculosis and can involve any organ. The Mycobacterium tuberculosis cell wall contains a complex wax which causes toxic effect. The toxic effect causes the dead cells to disintegrate but not completely digested by hydrolase. On gross examination, it shows this cheese-like consistency, soft, gray and friable. Microscopically, there is granuloma or with central area of caseous necrosis. The gross picture shows multiple nodules of caseous necrosis. You see this is one nodule, another set of nodules, and even more nodules here. The picture on the right shows what it looks like on microscopy. The central area shows lack of nucleus surrounded by areas of viable cells. The next type of necrosis that we'll see is gangrenous necrosis, which mostly is seen in the lower extremities and in the bowel. Gangrene refers to death of tissue due to hypoxia. There are two types of gangrenous necrosis. The first is dry gangrene, where coagulative necrosis occurs without the superimposed liquefaction. In this type of gangrene, the skin becomes dry, wrinkled, and dark brown in color. The second one is wet gangrene, which occurs when the necrosis is complicated by infection and invasion of neutrophils, causing liquefactive necrosis. The first picture shows an example of dry gangrene or necrosis in a part of the foot. The toes were involved in a frostbite injury. There is mainly coagulative necrosis from the injury, but there is no pus. The second picture shows wet gangrene. You can see pus over the toes. There is a liquefactive component from bacterial infection on top of the coagulative necrosis from ischemia. This patient had diabetes mellitus. The first picture shows an appendix and the second picture shows the large bowel. In both pictures, part of the appendix and bowel shows darker color at the arrow compared to the rest of the organ. This is a feature seen in gangrenous bowel. The last type of necrosis is fibrinoid necrosis, which is associated with immune complex deposition involving the blood vessels. This is seen when complexes of antigen and antibodies are deposited in the walls of the arteries or small blood vessels. These complexes will activate complement system and neutrophil activity, and the vessel wall undergoes necrosis. The immune complex deposits cause the bright pink and amorphous appearance as pointed by the red arrow. At this point in time, we have seen reversible and irreversible cell injury. And the difference between the two is in reversible cell injury, there is decreased ATP levels, which causes a lot of other cellular changes such as hydropic swelling, decreased pH, ion imbalance, and fatty change. In irreversible cell injury, there are more severe cellular changes such as severe membrane damage, which causes the enzymes from the cell to be released into the tissue. There's lysosomal rupture, which causes enzymes to be activated, and extensive DNA damage in terms of karyorrhexis, karyolysis, and pyknosis. We look at the systemic manifestation of cellular injury. When there is cellular injury, it can be manifested as fever, tachycardia, leukocytosis, pain, and presence of cellular enzymes in the blood. Fever is due to the presence of endogenous pyrogens, whereas tachycardia is because of the increased metabolism due to fever. Leukocytosis can occur because of infection. Pain is because of release of bradykinins and prostaglandin E2. Presence of cellular enzymes in the blood can point out to which organ is having the injury, depending on what type of material or enzymes that is released from the tissue cells. The enzyme release during cell injury include cardiac enzymes if the injury is involving the heart, such as myoglobin, CKMB, LDH, and troponin. Liver enzymes such as aspartate transaminase, AST, alanine transaminase, ALT, and alkaline phosphatase. And pancreas enzymes such as lipase and amylase.